I'm Coté from Radford University Biology Department, and my poster is on the microscopic crystals in aroid flowers of role in gamete protection. Nobody knows why plants make these crystals, even though they're found in plants of over 200 different families. But one idea that always comes up is they're there to protect the plant from being chewed on by herbivores. And of course, the most important part of the plant you want to protect would be the ovules and the pollen and the developing embryo, which represent the next generation. Now, the aroid family not only has a huge number of different kinds of crystals, but they also have some interesting pollination mechanisms. Some of them are pollinated by bees, like any other flower that we're familiar with. Some are pollinated by the flies that lay eggs in what look like dead, decaying corpses, and I'm confused by that. But more interestingly, in some species, the inflorescence closes up around beetles, which are trapped inside, and then spend the night having sex, eating the sterile flowers, basically partying, and then the next day they pick up pollen and take off to the another site. So our hypothesis was that different parts of the flower will be protected by crystals if they did in fact have a protective role, and other parts would not necessarily be so. And what we found was indeed that the female ovules are always packed with crystals around the ovules to protect them. The anthers with the unshed pollen again have crystals in the connective, especially in those species where they're fused into a synandrium. And in those plants that have tepals, the tepals are full of crystals which surround the whole flower. But the sterile flowers were the real puzzle, because you wouldn't expect them to have crystals since they're eaten by the beetles as a bribe, a food bribe. And yet they are packed with crystals in many of the species. Staminodia, they always had crystals. Some of them were packed with them, some didn't have quite as many, but they always had crystals in there, which doesn't make sense because they were supposed to be eaten as a food bribe. And we can't explain this, but in some of the species, there were a layer of crystals lower down in the staminodia, and the beetles would chew right through the staminodia and stop when they hit the crystals. So we know that the crystals are protecting some of them from being chewed all the way through and letting them eat the part they're supposed to eat. But we can't explain what's going on in others where the crystals would basically protect the whole staminodium. So the hypothesis does work for most of the flower, but we still have to figure out what's going on with the sterile staminodium. Thank you.